Clay-mation project by Kwon and Kevin Tal. Wilkins and Franklin used X-ray crystallography to produce an X-ray diffraction image of DNA. The spots and smudges were produced by the X-rays that deflected through the DNA. These images led to Watson and Crick's new discovery of the DNA structure. Watson and Crick carefully studied the X-ray images and discovered the width of the DNA helixes, which enabled them to deduct that there were two strands of DNA and that they ran anti-parallel from each other. Watson and Crick's semi-conservative model shows that when two strands of parental DNA separate, they each function as a template for a synthesis of a new complementary strand. The resulting daughter molecules will each have one old strand from the parent molecule and one newly made strand. Central dogma, a molecular chain of command with directional flow of genetic information. The central dogma starts with DNA replication. DNA replication begins in the nucleus where the chromosome is located. During DNA replication, the DNA consists of the leading and the lagging strand. The topoisomerase is an enzyme that helps relieve the strain caused by the helicase ahead of the replication fork by breaking, swiveling, and rejoining the DNA. The helicase untwists the double helix at the replication fork, making them available for replication. The single-strand binding proteins bind to unpaired DNA to stabilize them. The primase synthesizes RNA primers by using the parental DNA as a template. The RNA primers are short stretches of RNA, and this is where the DNA strand begins as DNA polymerase III adds nucleotides to the DNA. The leading strand is continuously synthesized in the 5' to 3' direction. DNA polymerase III has to discontinuously synthesize DNA on the lagging strand because of its 3' to 5' direction, and this creates a series of segments called Okazaki fragments. DNA polymerase I replaces the RNA primers, and the DNA ligase bonds the 3' end of one fragment to the 5' end of the other fragment. The lagging strand replicates DNA at a slower rate than the leading strand because of its direction of replication. The next step of the central dogma is transcription. The transcription process starts with DNA. The eukaryotic DNA contains a TATA box, which is a DNA sequence and part of the promoter, and helps transcription factors to bind to the DNA. The DNA also contains a transcription unit, which is a stretch of DNA transcribed into RNA. In bacteria, the promoter itself attracts the RNA polymerase to attach to the DNA. However, in eukaryotes, the DNA needs the help of transcription factors for RNA polymerase to attach to the DNA. During the first stage of transcription, called initiation, the RNA polymerase binds to the DNA and RNA synthesis begins. During the second stage of transcription, called elongation, the RNA polymerase moves down and unwinds the DNA and elongates the RNA transcript. The RNA transcript moves in the direction of 5' to 3', and it gets longer as the elongation process continues. The third stage of transcription is termination, and the RNA transcript is released and the polymerase detaches from the DNA. RNA processing modifies the transcribed RNA to produce an mRNA ready for translation. During RNA processing, a 5' cap and a poly A tail is added to the RNA, and they facilitate the export of mRNA into the cytoplasm, protect it from degrading, and help ribosomes attach to it in the cytoplasm. Also, exons and introns are designated. SNRPs, or small nuclear ribonuclear proteins, are located in the cell nucleus and consist of RNA and protein molecules. They combine with other proteins to form an assembly called a spliceosome. The snRNA of the spliceosome base pairs with nucleotides at specific sites of introns, and the spliceosome cuts out the introns from the mRNA while splicing the exons together. Now the mRNA only consists of the 5' cap, exons, and poly A tail. The third step of the central dogma is translation. After transcription, the mRNA has formed and it now leaves the nucleus and enters the cytoplasm. A small ribosomal subunit attaches to the mRNA and it recognizes a sequence just upstream of the start codon. An initiator tRNA with a complementary anticodon of the start codon on the mRNA base pairs with the start codon, and it carries an amino acid. The large ribosomal subunit comes and binds to the mRNA and the small ribosomal subunit, which completes the translation initiation complex. The initiation of translation is complete. During elongation, another tRNA carrying a new amino acid forms a peptide bond with the growing polypeptide. The ribosome moves the tRNA to the A site, P site, and E site where they are ejected. The cycle repeats. During termination, the ribosome reaches a stop codon and accepts a release factor. This causes the hydrolysis between the tRNA and the last amino acid. The two ribosomal subunits and other components of the translation complex completely dissociate. Protein synthesis regulation. An example is alternative RNA splicing. The DNA has certain genes that will later become exons or introns in the mRNA. After the transcription of DNA, which creates a primary RNA transcript, the alternative RNA splicing can create different mRNA molecules depending on which parts of the RNA are treated as exons or introns. Missense mutation. Substitutions that change one amino acid to another one. An example of missense mutation is sickle cell disease. A normal functioning hemoglobin DNA should look like this. Transcription of the DNA would produce an mRNA that coded for a certain hemoglobin protein. In this case, it coded for glutamic acid, which resulted in a normal hemoglobin. Mutation. The mutated DNA has an A instead of a T at the top. As a result, the mutant mRNA has a U base instead of an A base. This results in a translation of the wrong amino acid, valine, instead of glutamic acid, and the sickle cell hemoglobin is formed. The amino acyl tRNA synthetase facilitates the creation of amino acyl tRNA. The amino acid binds with ATP on the active site, and the ATP loses two phosphate groups to become AMP. The appropriate tRNA replaces AMP and bonds to the amino acid. This results in the creation of the amino acyl tRNA, which is released by the enzyme.